So in getting right into the visual system, we're really here starting a new module. If you think back to the introductory lecture, you'll realize that we said we were going to have an introductory module, and then the second module that encompasses the next two quizzes is all about sensory systems. As I said, this chapter that goes with the visual system is pretty dense, but for some people, they really like this material. It tends to be tough, but some people enjoy it more. And that's mainly because for some people, concepts like neuroscience methods are really abstract, but when we start to apply things and we can tell a story about what they're doing, some people attach to that a little bit better. So only you know yourself and you'll be able to decide. But we're going to spend the next two lectures covering the visual system and using it as kind of a model for what other sensory systems are doing. So what should you know for each of the sensory systems? Well, some main things that are going to come through is basically an understanding of how the sensory system works. And this is a process called transduction. So how do we take the stimulus in the environment, like sound or light, and how does that get turned into a signal for the nervous system? So this includes, like you see here, the basic terms and context and concepts that are involved, and also any of the different neurons that participate. So as you're taking notes, this would be something great to focus on. Another thing that you should know a little bit is what kind of the main pathway and circuits are. And this should be incredibly easy with Open Book and Open Note. So you need to look at these pathways and circuits and understand kind of what the function of certain structures are as a third piece of information to know, and then how these connections are organized. So a flow diagram would be great here, including the functions. So A connects to B connects to C, and some of this might be at the single neuron level for things like transduction, and then other parts of it might be at the whole brain level and for functional roles and what we think that certain brain structures do. The role of the sensory system, you all know. This is actually one of the easier concepts to grasp because this is something you all experience in a way that is very easy to put a finger on. Our sensory systems inform the brain about what's going on around us, and it helps us to use information to guide or change behaviors accordingly. And the second piece that you might not think about so much is that the sensory feedback is also important for kind of modifying what's going on. So this can be things like, regulating body temperature, and there's a lot of these more subliminal pieces, or the idea of proprioception, that we're all kind of aware of where we are in our own space, so we don't walk into things all the time, and yet a lot of that's happening at a subconscious level. So starting from the most basic definition, a sensory stimulus is just some kind of energy that can activate a receptor on some neuron in the body. And when we're talking about these sensory receptors, then they're not quite the same, and you're going to see this. They have all kinds of different ways that they accomplish this process of transferring energy into a neural signal. So when we're talking about these receptors, we're actually talking about specialized kinds of cells that receive energy from the environment. So in a way, the whole cell in this case is a receptor. We're not talking about one little protein channel that's on a neuron. We're talking about this entire neuron whose job it is to receive energy from the environment. And this can be mechanical stimulation, which happens in touch. It can be chemical stimulation, which happens in taste and also in smell. It can be thermal stimuli, which happens in touch and in a couple of other places. It can be light or sound waves for the visual and the auditory system. The important concept that unites all of these is that all of these different things that are being turned into a neural signal are a form of energy. So they're physical in some way, they're measurable. You all know that light has wavelengths, and sounds have frequencies, and touch has force. So you put all those things together, and that's what our brain is doing these calculations on. And this brings us to the concept of what's happening. So we talked in the slide of what to know, I mentioned this word transduction. So transduction is the process of taking these signals from the environment, these energies, and turning them into a neural signal. That process is called transduction. And a very important thing to know is this concept we call the law of specific nerve energies, which is basically that all of the different receptors in your body respond to one kind of energy. And so the receptors are specialized in order to process that kind of energy. And this produces the sensation we have, but also the perception of different things in the environment. So if you remember back to the history lecture, I usually do this there, but I also do it here as well. And if you don't, you can do this again, where if you take your finger and you press onto your eye for just a second, not too hard, please, and you release it, eyelid maybe is a better way to say this, but pressing on your eyelid, it will cause you to see kind of like a halo for a second, right? It'll cause you to see some little bit of color or something like that. And that has to do with the fact that our eyes are used to responding to one specific kind of energy, and they send a signal to the brain that matches that kind of energy. So if we give them a different kind of energy that can still affect those neurons, hey, we still perceive something that's kind of like light. So each of these receptors responds to one kind of energy. It's very specific in what it can detect. We call this tuning. And our perception is what we think of as kind of the conscious experience. So that can differ from person to person. So the level of the energy might be the same. For example, the light, would be at a fixed brightness. And yet our perception of that light can change. For example, if they're, one of those people that's not in physio-psych that goes out normally at Rutgers and parties on the weekends and ends up with one of those hangover things, might perceive the same light as being really painful one day as compared to a normal day when they didn't, let's say, go to the Golden Rail for 6 hours the night before. So those are the kind of things that that can change, right? Our sensation does not change, but our perception does. The sensation is just the energy coming in. The perception is our experience of how that's coming through. Another thing that's common to all of these systems is the idea of a map. So we call this topography, right? Just like we would say on a map, but all of the different sensory systems we're going to talk about have some sort of map to them. In some of them, it's very easy to discern. For example, in the cochlea, frequencies of sound are represented almost in a linear way, where low frequencies are at the apical end of the cochlea, and at the basal end we have high frequencies. And this transfers directly to also a map within the auditory cortex. The same thing happens for the eye, where basically the way that the cells are arranged in the eye are kind of like pixels on your television, that you have kind of a left to right and an up and down, and the cells in your eye that are capturing energy in different parts of the visual field or the kind of the space around you are mapping back onto the brain in basically a similar kind of location. And then some are a little bit less intuitive, like a somatotopic map, where our bodies, some places are more or less sensitive, and so we have a map of all of our different body parts that, there's a part of your brain that only perceives fingers and another that only perceives toes. But there's a map nonetheless of all of those areas. And probably the most confusing are the ones for taste and smell, because the map isn't necessarily like physical location, as you might think of for most things, but instead has something to do with chemical structure. The next thing that we need to consider is sensory resolution. The basic thing you need to know here is that the density of whatever sensory receptor we're looking at is directly related to a few things. The first is the number of connections coming from whatever the sensory organ is all the way into the central nervous system and on their way to the brain. The second thing is that the number of neurons on each level going then from that sensory neuron through to the brain also will match that. So this means if we tend to have, you're going to see there's usually 2, if not 3 or 4 connections before we hit kind of the primary part of cortex responsible for vision or smell. At least in most of these cases, we have a couple of steps. And so this means we have more neurons at each level related to whatever information was coming in from, let's say, the eye. And then the last thing is this means that the size of whatever cortical area for primary visual or somatosensory cortex that we might be looking at is also going to be bigger. The easiest example of this is if we look at what goes on in the somatosensory cortex, which is responsible for touch. And the way to look at this here is that different parts of the body are represented kind of by their size. And so the cortex related to fingers and the hand, which we all know have pretty fine resolution. You can detect really subtle touch onto your hand, for example, or onto your face and lips. Those areas also have more cortical space, more total tissue devoted to them, whereas things like your torso don't have nearly as much related to them in terms of cortical size. So put very succinctly, the higher density of sensory receptors, the higher the resolution will be for that sense in the brain, and the lower density of receptors, then kind of the lower resolution or ability to detect things that we have. Another thing that's really important to talk about here is kind of immediately breaks everything we talked about in terms of the rate law. So sensory systems are a bit of an exception to the rate law, and instead they have signals that we think of as being analog. The best way that I can describe analog is the volume on, let's say, your car stereo or something like that, where if you twist a knob, you can go from low volume all the way up to high volume. And that's the way that these sensory systems work, because they have to match what's happening in the environment. So we have stimuli that can be tiny all the way through really bright or really loud, and we need to be able to detect that. So what happens in these sensory systems is that small intensity stimuli produce small receptor potentials. And eventually, usually a couple of neurons down the line, this translates into our rate law, where we will have fewer action potentials. Whereas really loud or bright or high intensity stimuli will create higher receptor potentials and therefore eventually translate into a higher rate of action potentials. And just as a reminder, the rate law is basically the idea that if we have, let's say, some kind of a weak stimulus, in this case sensory, then it will cause the neuron to fire fewer action potentials. And if we have a stronger stimulus, it will cause the neuron to fire more action potentials. So again, just as a reminder for those of you who may not remember that because we've only gone through it once. Okay, so it's really important that we talk about the structure of the visual system. The visual system is designed to detect wavelengths of light. And so light is part of the electromagnetic spectrum, and what we detect with our eyes is basically something from around 400 nanometers. So this is kind of a small unit of measurement, but notice that the unit here is meters. There's an actual physical wavelength to this energy, to somewhere around 700 nanometers. You might see across books or references that this is people will say this might be a little bit more or a little bit less. Don't worry about it. This is an approximate range. And as always, some humans are better at detecting things that are at the ends of these spectrums than others, based off of their individualized biology. However, this is something that can also change across species. So it's not necessarily unique to us, but there are some animals that see fewer colors, and there are some that can see ranges of color that we can't see, for example. In terms of the anatomy of the eye, this is also very important for how we get each of these signals through. So the main parts of the eye that you need to be aware of are the pupil, which is used to admit light, the lens that we use to focus the light, and then of course the retina, which we'll see in a second here is really composed of a few different components. That's the place where we finally transduce the light. And I think many of you know that your pupils can change in size in order to help control the amount of light that we emit, and the lens is probably more of an active process than some of you might think about. So this all helps to change the way that the signal reaches the retina, and of course then how it's transduced. And the one thing I want you to all start thinking about is that if you stare straight ahead right now, your visual field is basically a grid. Of course, it's coming across two eyes, but you have things that are kind of on the left side of your visual field, the right side of your visual field. There are things that are up and down. And so it's a good idea to start thinking about the visual field as like one big grid across a piece of graph paper. And this is mapped back onto the retina in pretty much exactly that manner. Let's not worry about kind of all the weird flips and other things like that for the moment, but there's a one-to-one relationship between what's out in the visual field and what's being mapped onto the retina. Take it at face value for now. The basic flow of information is that we have light coming in, and that happens somewhere about here. And I've been teaching this class for long enough to know that there's going to be a question that comes up here just like in a second. So I'm going to cut it off at the pass. So light comes in at the ganglion cells, but the photoreceptors that are responsible for detecting it are way back here in the deepest layer of the retina. So yes, light starts out and it passes through two layers of different kinds of basically sensory neurons before it finally hits the photoreceptor. I've never really found what I feel like is a great answer for why this happens. The only thing I can think is that these other two layers of cells help to dampen the light a little bit, and this is just something that developed over evolutionary biology. So keep that in mind. There's this kind of backwards thing going on here, but light comes in and it passes through these ganglion and bipolar cells before it hits the photoreceptors. So talking about each of these layers for a second, bipolar cells were given their name because they have two poles, just like the Earth, branching off in two directions. And their job is really simple. It's just to connect these ganglion cells and these photoreceptor cells. They're basically a bridge. That's what you need to know. The ganglion cells out here in this shallowest layer of the retina, right, exactly where the light first hits the retina, their job is to send long-range projections to the brain. And then we get to the photoreceptors. These are the primary cells of interest for us in terms of transduction. And as I think many of you know, they come in two classes because someone who looking at them under a microscope once upon a time was really creative and said, hey, these ones kind of look like a big rod. Let's call them rod cells. And hey, these ones look like a big cone. Let's call them cone cells. End of meeting. So these have very different properties for each of them, but these are the two main classes of photoreceptors that make up kind of this sensory portion of the eye. Something you may not know, because I think probably most of you have heard of rods and cones, is that rods, which are actually the ones that are sensitive to dim light and provide colorless information, these actually outnumber the cones. So there's about 120 million rods in your retina and only about 6 million cones. And in spite of this, somehow the cones provide really high resolution information. So this seems a little bit backwards from what I just told you a minute ago, where there's normally kind of a one-to-one relationship between neurons and resolution. So why is this? How do we get fewer neurons providing better visual information? And the truth is that all of this reconciles just one step down the road. So the first thing you need to know is that every cell participates in some part of our receptive field. So basically this means that every cell receives information from just some part of the visual field. This could be the center or this could be the edges. And so we call the edges the periphery and the center the fovea. And so most of the information, as you all know, the kind of the best and most in-focus information comes from the fovea, right in the center of the eye. And things get a little bit more difficult to detect out in your periphery. And some people have a little bit better peripheral vision than others, but for all of us, the peripheral is not quite as good. So an important feature of this center, or this fovea, is that the cones get really densely packed in there. So this means that despite there being fewer of them, they actually get more of the more important information. And the rods are found more densely in the periphery of the retina. So that's a starting point for how this happens. The second thing is that we have some different properties for how these interact with the bipolar cells. And the basic idea here is that cones have a one-to-one relationship with the bipolar cells, which means that they carry more weight. So the signal from one cone pretty much has direct access to one bipolar cell, one ganglion cell, and this means that the number of connections for these reaching the brain is 1-to-1. On the other hand, the rods do not have this 1-to-1 connection. And in fact, they can be more like 10 or 20 or more to 1. So there's some really interesting things that this allows to have happen. So this means if the cone is able to send a signal to a bipolar cell and activate it, then that signal is given kind of a fast pass to the brain. Whereas the rods, they share this signal. And so this means that the message to the brain from the cones is more precise. However, this also means that the cones fail under certain circumstances. And the circumstances in which they would fail would be if there's not enough light getting to the cone. And instead, this is where the rods do a really, really good job, because now they're using teamwork. They are 10 or 20 to 1, and that means if their job is to send a signal that affects this bipolar cell, and there's very dim light, but you have 10 rods combining together to send a little bit of information about that dim signal, then that means that there's now, from the same amount of light, there's now 10 times the neurotransmitter being released onto these bipolar cells, for example. So in that case, they actually perform much, much better because of this idea we call convergence, where many of them converge on one bipolar cell. This allows them to perform better under dim light situations, but it also means the information from any one rod is diluted down. So if I have a visual stimulus come in to one rod, it's basically the information it gives to the bipolar cell is now divided by 10 or divided by 20, and it doesn't have as much, it doesn't provide as much visual acuity or as much resolution in terms of what gets to the brain. So it's kind of a trade-off, and that's an important trade-off to know. Now, just for a second, because I know if you're reading through your book, you're going to stop and you're going to hit a section about these horizontal amacrine cells, and you're going to say, we talked about all the cells in the eye and the retina, but we didn't talk about these. What I just want to tell you is, at least in terms of what you should know, the horizontal cells act as a bridge, kind of at the connection between the photoreceptors and the bipolar cells. So their job is to pass information. You could think of it as like horizontally between photoreceptors or bipolar cell connections at this level. And the amacrine cells do this at the level where the bipolar cells contact the ganglion cells. So this allows kind of crosstalk between nearby photoreceptors or crosstalk between nearby ganglion cells. That's the important thing to know here. So as a quick summary of this section, we have cones that provide high visual acuity because they are found very densely in the fovea. And this means that because there are so few of them, we need high levels of light in order to get that acuity out of them. They also provide color information. And so that's kind of the main features of the cones. Whereas the rods are found more often in the peripheral retina, where not as much information gets in the first place. And they're also very sensitive to low levels of light because they have this kind of converging property, but they only provide monochromatic information, so black and white or grayscale, whichever way you want to think about it. And also because their information is kind of divided in terms of what gets into a bipolar cell, they provide poor visual acuity. Okay, so how do we transduce a visual stimulus? Now we kind of know all the players. What happens? So when light comes in, and remember it's coming in from out here, you can see the direction passing back here, it hits photopigments in the eye. Okay? So what happens when it hits these photopigments is that there's a chemical reaction here. So the photopigments that are in the photoreceptors basically have two parts. There's a protein called an opsin, kind of like the channel rhodopsin that we just talked about for optogenetics at the end of chapter 5. And there's also a lipid called retinol. And so when light hits the eye, the rhodopsin and the retinol separate, and this basically is an internal trigger, kind of like when we talked about the metabotropic receptors, that basically causes the photoreceptors to change the way that they release neurotransmitter. And for most of them, this ends up hyperpolarizing them, right? So it actually decreases their activity. And what ends up happening then is that the bipolar cells kind of reverse the signal. So they end up, when the photoreceptors are when the photoreceptors are hyperpolarized, they release less neurotransmitter. And then we have the bipolar cells that are depolarized, and they start to release more neurotransmitter. Now, in the case of both the photoreceptor and the bipolar cell, this release of neurotransmitter is, notice I'm saying release of neurotransmitter and hyperpolarized and depolarized. I'm not saying action potentials, because it takes all the way until these ganglion cells for us to finally get this into that rate code. So this is what I was talking about just a minute ago. This is not, the photoreceptor can go from releasing 1 unit of neurotransmitter or 1% of neurotransmitter all the way up to 100%. So it scales evenly from zero light all the way to bright light. And the bipolar cell is kind of the same way. It just inverts the signal. And then finally, the neurotransmitter released onto the ganglion cell, if there's a lot of neurotransmitter release, this translates into a higher rate of action potentials. And if there's very little, then it translates into fewer action potentials. Another thing I want to point out here is that if light shuts down these photoreceptors, then in a way, you could think about what they're actually detecting. The thing that activates them is darkness. So in a weird way, they're kind of like darkness detectors. Just to put that all together. So another thing I think I should probably mention here, just because I think it's important, is so we have this chemical reaction, rhodopsin and retinol separate. This triggers this hyperpolarization. The neurotransmitter being released here is glutamate. So these hyperpolarized photoreceptors release less glutamate, but that causes the bipolar cells to then release more glutamate. So glutamate is an excitatory neurotransmitter, at least in general terms. So that means that more glutamate release here means more activity in these ganglion cells. So that's one piece I knew I was forgetting in the back of my head that's important as we go through. Another thing that's really important for me to mention is that there are actually two types of bipolar cells. So I gave you just the one example, but we actually have bipolar cells that differ in the way that they respond. And this is going to become incredibly important as we start to talk about things like how we detect edges, where one thing in our visual field stops and another one starts. So bipolar cells that are what are called on-center are actually hyperpolarized by glutamate. So more glutamate means that they're going to shut down. And the others that we call off-center cells are depolarized by glutamate. So they can have kind of two opposing responses here. And the fact that we have both of these is very, very important. In both cases, depolarization of this bipolar cell, like you see in the example here, is going to increase glutamate release and relates to more action potentials firing. And the ganglion cells then follow the rate law, right? So more glutamate here means more action potentials, and so on and so forth. So what this really means is that the bipolar cells can kind of have opposing responses to where some are really detecting light and others are detecting dark in a roundabout way. And you can probably imagine a little bit of how that might come together to create something like edge detection or even movement detection. So to put that together, it looks something like this. So if there is light in the center of the responsive field for this photoreceptor, right? So if imagine somebody shining a light into the center of your eye, for example, in this case, just to make it easy, then the cone here would be hyperpolarized because the light is on center. And now the glutamate release here is decreased. And this happens the same no matter what. So no matter what, if light hits the cone directly, then we have less glutamate release. But then that means that there are some bipolar cells where they're going to increase their neurotransmitter release and they're going to cause more firing in the ganglion cell. And there's another class of off-center ones that are going to be hyperpolarized, decrease their glutamate release, and cause less firing in a ganglion cell. Now, in another circumstance, if light comes and it's not right on the center of this cone, then these neurons are now going to flip jobs, where again, the cone is going to do the same thing. The cone is going to be depolarized because it's not receiving light in this case, so it's doing a darkness detector kind of thing. But because of that, there's more glutamate that's released, and these on-center bipolar cells that had fired before now all of a sudden shut down, and their ganglion cells shut down. And the off-center bipolar cells now increase their activity because the light is off of the center of this, and then their ganglion cells increase activity. So if we try and really put this all together, I know this is an incredibly complicated concept, and we're going to come back to it a couple of different times. So let me take all of what I just said and simplify it. And then what I'm going to encourage you to do is to pause the video and go back through the slides or rewind a bit until you get this. This is a concept I know a lot of people struggle with, and we would probably have some questions if we were doing this in person. What we've done here is we've taken neurons and we've given them different jobs. So I have one neuron that is kind of responsive when something is dark, and another neuron that's responsive when something is light. So let's just take our visual field here, right? So we know we have bipolar cells that are responding to different parts of the visual field, and it's probably important here to say that these can have a little bit of crosstalk with each other. So they kind of know what's going on with the cells nearby. So what this means is that if I have any sort of a difference in brightness, let's just take this dark black portion of the soccer ball here in center, and then the grass right next to it that's green. So let's call this the surround. So let's say this part of the ball is darker than the light part here. Well, one option would be that I could have neurons just detect when something is dark, right? And that would mean I would do a great job of detecting this, but they would be firing kind of to the degree of darkness here, and that gives us poorer information. So instead, we give two different types of bipolar cells different jobs, where one is responsible for saying this is lighter than that, and the other one's responsible for saying this is darker than that. And by having both of those and having kind of redundant information, then we're actually able to use those separate classes of cells, and then as information gets back into the brain and the cortex, we can use those to compute exactly how different these are. So you can think of it as kind of like some visual arithmetic, where I can say, well, this is firing at 5 spikes per second of brightness, and this is firing at 10 spikes per second of darkness, and we can compute the difference between those two later. Whereas if we only had kind of lightness detectors or only had darkness detectors, they would not do nearly as good of a job. So in a way, by having these opposite responses, we're able to do this mental arithmetic and enhance contrast between things. So again, weird concept, but go back through it a couple of times. Don't be afraid to just step back. It'll be important for you understanding this later. And we'll go through this in the second part of this visual series as well, as we hit other important pieces of information. Okay, so now that we've gotten through this complicated concept, let's go back and simplify for a minute. Just talk about how visual information reaches the brain. The first step in information getting to the brain is that the axons from all of these ganglion cells across the entire retina, they come together to form the optic nerve. And the optic nerve is then going to be the place where information starts to head back towards the brain. The optic nerves then synapse on neurons in the part of the brain called the lateral geniculate nucleus. So if you're remembering what I said very early on in the lecture, this is the place where you should be starting to produce some sort of a flow diagram. Okay, ganglion cells, lateral geniculate nucleus. So a really important feature, though, that you're going to notice here is that the information that reaches these cells, these neurons in the lateral geniculate nucleus, is related to a certain part of the visual field. And so I think there's a bit of a misnomer here where people say that, information from the left eye reaches the right side of the brain and the right eye reaches the left side of the brain. And number one, it's not a perfect relationship anyway. But number two, it's not left and right eye, but it's actually the parts of the visual field. So on the right side of both retinas here, you'll see this green pathway. And so this is, if we draw this diagonal over here, it's actually the place where the light would pass perfectly through the lens. So in some ways it kind of makes sense, right? This is the perfect diagonal through the lens. And so this represents the left half of the visual field. And yes, and of course, this does all then group together and come to the right lateral geniculate nucleus and then back into cortex. And the same thing is true of the right side of the visual field. This comes to kind of the left side of the eye where the light directly passes through the lens and it crisscrosses, and this ends up passing back then into the lateral geniculate nucleus as a group from both of the right halves of the visual field and back into cortex. So from the lateral geniculate nucleus, the information goes back then for each part of the visual field into the primary visual cortex, sometimes also called V1, or the striate cortex. Your book likes to use striate cortex a lot. I think it's easier for you all to remember primary visual cortex, or V1, one being first or primary. So that's the terminology I like to use, but your book used to striate a lot, so just so that you don't confuse them, I'm going to say them all here just once. Okay? And so again, take note of the fact that the right side of the visual cortex is getting information from the left visual field, not the left eye, but the left side of the visual field. So this is where that idea of the crossing comes from. And this information and all of this crossing happens at this place we call the optic chiasm, where the information from the optic nerves crosses and kind of goes, some of it at least, to the opposite side of the brain. That's this kind of crisscross pattern you see here. So you can see left side of visual field in the case that I've highlighted here, comes to the right side of both eyes, the place where it perfectly passes through the lens and hits to, goes to the right lateral geniculate nucleus and back to the right primary visual cortex. And then information from the primary visual cortex is actually pretty raw. So you might think this is where a bunch of processing happens, but at each step of what's happening here, right, and starting with the eye and then going to the lateral geniculate nucleus and primary visual cortex, there's a little bit more processing going on at each step. And the final place where all of this goes is the secondary visual cortex, sometimes called V2, or visual association cortex, or the extrastriate. So your book uses way too many names. I apologize for that in advance. I didn't write it. But this secondary visual cortex, which is what I'm going to refer to it as, is the place where we finally kind of get to a full picture of what is happening in the world in front of us. So let's talk a little bit about some of these visual regions. What are they doing? The lateral geniculate nucleus has 6 layers, and it's in a part of the brain we call the thalamus. So the information that's being processed depends on the layer of the lateral geniculate nucleus. And I want you to think of this as kind of like a sorting system, right? The LGN, or lateral geniculate nucleus, is a sorting system. The first segregation that happens in the LGN is by eye. We just saw that we have information coming from both eyes, but the same part of the visual field, whether left or right. and it goes to the LGN. But that information stays kind of separate, which is really important for certain kinds of calculations that the visual system does. So we have layers 1, 4, and 6 processing information from the contralateral eye, or the one that's on the opposite side of the face from whatever lateral geniculate nucleus there is, and ipsilateral means same side. So we also have information coming from the ipsilateral eye to layers 2, 3, and 5. So it's not quite even or odd, but there's equal representation. There's 3 layers dedicated to the contralateral and 3 to the ipsilateral. And then we have 3 different sets of layers. So we have layers 1 and 2, or what we call the magnocellular layers, and this is why this doesn't end up being a perfect even and odd. And so these first 2 layers are dedicated to movement. And then layers 3 through 6 are what we call the parvocellular layers, and they're related to color and detail. So again, think of this as a filtering system. We've started to filter information about movement, which you might imagine comes more from rods that are better at detecting contrast and light and dark differences, whereas color and detail is more coming from the cones. And then between all of these, in something that oddly your book kind of poorly represents, we have these coniocellular layers, and these are important for processing color information. When our information here coming in from the LGN hits the primary visual cortex or the striate cortex, there are 6 layers, but the primary input comes into layer 4 from the LGN. Layer 4 is mainly getting from magnocellular and parvocellular, but just for the sake of simplicity here, layer 4 is where the major input comes in. And we're going to talk about all kinds of specific processing that happens. That's kind of the focus of part 2 of the visual system lecture. But for the sake of simplicity, again, the visual information is sent to layers above and below for analysis. And I think it's important to say that the information is kept in some ways organized, just like it was in the LGN. So the visual system, when things get to the primary visual cortex, information related to movement is going to be processed together. And so the primary visual cortex might send movement-related information down one path and then information about color down a completely different path. So this information stays organized, and it's not a complete picture of what's happening yet. And the primary visual cortex starts to do some combination and analysis, but it takes all the way until the secondary visual cortex for that kind of final picture again to take place. So we start to combine certain pieces of information, but some of the information still remains segregated. Part of the reason that we know this is that there were some really early studies that were done, and they're very interesting studies that show the kind of different features that these cells in the primary visual cortex respond to. So let's look at that first before we move on. So here's a video from Hubel and Wiesel, who would end up going on to win a Nobel Prize for their work in the visual system. So I want to show this to you, and then I'll talk to you a little bit about kind of a few things that we see here.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen and nothing seemed to work. And rather by accident, one day we were shining small spots, either white spots or black spots, onto the screen, and we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina. And every time we did that, we'd get a response.
So a really interesting thing here. First, I always like to point out that Hubel and Wiesel, they start out with these experiments, and like much of what happens in science, they come across this happy accidental discovery. He says, we were starting out and our first experiments were complete failures, right? And yet these are Nobel Prize winners. So observational experiments are so incredibly important. And the other thing that you'll notice here is that they find that this very specific line that's moving is what ends up activating these neurons that they're recording. So you could hear the pops, the little brr, 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 that was going across in the video, and those were actually neurons firing action potentials. That's a speaker that's playing back the action potentials. They're very fast, so it's hard to hear even each of the individual ones, but at some points you do. So what Hubel and Wiesel discovered, and we're going to refine this a little bit as we move forward, is that cells respond to very specific information. And this is an idea we're going to come back to over and over again, but the idea that certain neurons are tuned. And in the case of the cells they're recording, the line, the orientation of the line matters. And so part of what some neurons respond to is that line orientation. So in this case, a cell might fire the most action potentials to a bar that's completely vertical. So this up and down bar, you get the highest rate of action potentials. And things that are close by, tilted maybe one way or another, will cause fewer action potentials to fire. So they're close to the tuning, but they're not spot on. And the further and further we get away from that, all the way to a horizontal line, the fewer action potentials that we're going to see when that stimulus passes by. And the other thing that they unknowingly show you in that video is that the part of the receptive field matters. So the reason that they were getting that tuning is not only because they had a line that was in the right orientation, but it was a line that was in the right orientation in the right part of the visual field. And so when that slide hits the center of where they had, in this case, that anesthetized cat's eye facing, then that line was the right orientation and the right place in the visual field to get that neuron to fire a bunch of action potentials. And when they move it across, you see it fire fewer and fewer at the beginning and fewer and fewer at the end because they get outside of that range. So all of those details come together, and you can add on top of that other features like color and so on and so forth to create kind of the way that neurons in the primary visual cortex are tuned. So some things that we've learned from this is that there are a lot of features that neurons in the primary visual cortex respond to. They respond to texture, they respond to line orientation, they respond to the direction of movement, and they can also respond to things like patterns and spatial gradients, which we actually know a whole lot more about now. And again, we're going to talk about how Hubel and Wiesel were right, but not quite right next class. So getting back to this idea then of mapping things into visual space, right? I've been telling you this whole time, and it's a little easier to visualize here. You should start thinking of the visual field as kind of a grid. And what ends up happening is that information in the center of the visual field is the most acute, and information near the edges is less acute. And this, we have information coming from the left side of the visual field to the right side of both eyes, either the left or the right eye. You can see this blue pattern here, and this goes back into the lateral geniculate nucleus, and then back into the striate or primary visual cortex. And one thing I want to point out is that if we then look at how this is set up, you can see this information in the center of the visual field gets more space in the cortex, right? It's higher resolution information for the eyes, and the brain sees this as higher resolution as well, because there are more neurons devoted to the fovea, and that means that we get better information all the way back to the brain, and that means more neurons across the entire track are dedicated to that. So one way that we think of this information being represented is in terms of modules, and I'll tell you what I mean by that in just a second here. But an important thing to know is that this information coming back into the striate cortex is still broken apart into kind of specific features. this idea of kind of organizing or sorting that I've been talking about this whole time. And this is done by different modules in the primary visual cortex. So the information that's here, all the information is coming through, but it's separated and sorted in a way to where it hasn't been put back together yet. You could think of this as, you know, the different bags in a kit of Legos. They're all there, but they haven't really been combined in the right way to create that full creation yet. These modules I'm talking about are just a way of thinking about this sorting that I've been discussing for now half of this lecture. So the primary visual cortex or striate cortex has different columns, and each of them is processing a different type of information. So we have columns that are responsive to the orientation of lines, and each adjacent column responds to something that's shifted by, let's say, about 10 degrees. And then we also have columns that are responsible for the input from a certain eye. So we would have the input from the left eye dominating in one part and the information from the right eye dominating in another column. And so, of course, that means we have the orientation, sensitivity, and then that's represented across the left and the right eye, and maybe multiple cells representing the same orientation. And then a third piece of information that comes in here is information about color. And so these come into regions that we call CO blobs. And I don't want to go too much into this because I think it's more confusing than anything, but there were just early studies that showed that these can be stained using a stain called cytochrome oxidase. So that's where the CO comes from. But what you should know is that these areas are important for responding to color. And the areas that don't stain for this do not respond to color. So now you have to think about putting all of these different pieces together. We have left eye and right eye. We have, of course, left primary visual cortex and right primary visual cortex. And then we have all these orientation columns. We have color versus no color, and so on and so forth. And if you put all of these possible permutations together and multiply them, this is where we finally get out to the around 2,500 different groupings of information. And you have to add on to this some of the other things that I'm not even mentioning here, like whether we have the bipolar cells that are responding to light being on the center versus off of the center, and so on and so forth. So you can kind of put all these pieces together, but this is just a way to represent how the visual system is organized. It's not something you need to know all of the details of. I'm not going to ask about one specific module and how it might respond. You should just know that the information comes in a way that is mapped and organized according to all of these different features. The secondary visual cortex, which is kind of the last stop that we talk about, is where we start to combine these features, and we'll spend way more time on kind of some of the ways that the information is combined the next part of this lecture, but this is where we group information together that allows us to perceive objects or scenes, and there's kind of a hierarchical structure to the way this information comes in. So we're talking about this organization of the system in a very general way this class. So layers that are closer to the primary visual cortex receive information that is more consistent with the modules, right? So it's information that's kind of organized, but in segregated into different categories. And then as we go through the layers of the secondary visual cortex, the information becomes more and more and more processed. So we might go from having separate information about brightness and contrast that's kind of coming from rods and color information. We might combine that together to process something that is colorful but has really stark contrasting edges. Or we might also use that to have information about what the features of color are for all the features of a person's face, and separately information about movement, kind of having different parvo and magnocellular information, and combine that into what we perceive as a person moving and running. So the secondary visual cortex is where quite a bit of this magic happens, where we finally compute all these pieces into our perception. So what happens after the secondary visual cortex gets a little bit complicated? And yet visual researchers have done a very good job of simplifying this. The way that we think about the processing after this is that information flows in two streams. And it's very important that you know these streams and what their jobs are. This is something we talk about for function. And in case you haven't caught on, when I say it's very important that you know X, it usually means that there's probably going to be some information that I'm going to be testing about this. So for those of you that have stuck through all 59 minutes we've been here so far, thank you. And here is your hint of the day. The dorsal stream involves information about where an object is. And this, all of these pieces of information go on to kind of higher order association areas where we start to combine information about visual stimuli with auditory stimuli and all kinds of things like that. So this dorsal stream is known as the where stream because it helps us to process where objects are in space. The second stream is called the ventral stream. So this involves object identification, so what an object is. And those are two very different pieces of information that are equally important. I need to both know where things are in the environment, and I also need to be able to identify them. And it turns out the brain processes those kind of information differently. There's so many different ways that we know that these two streams exist. We have functional magnetic resonance imaging, we have recordings in cats and in primates, and we've done many different things, but there's something also really powerful about developmental studies. And so one of my favorite studies in thinking about the dorsal and the ventral stream is essentially one where we see that kids at some point in their lives are really unable to connect the information between the two streams. And this tells us a lot about how the brain needs to make these associations and connections in order to fully understand the environment around it and be able to use the sensory system to its fullest. So with that, what I'm going to leave you with today is a video with some cute babies to send you off and start you off in the week in the right direction. And I hope that you'll find this one as both informative and funny as I do, and just a nice change of pace.
At Judy Delocha's lab at the University of Virginia, while studying children's understanding of scale, researchers discovered a very surprising phenomenon involving self-awareness, a crucial step toward becoming a social being.
All right.
2.5-year-old Madeline is invited to play in a room full of large toys. At this age, she already has a sense of herself and would have passed the shopping cart test with flying colors. She's given hands-on experience with each toy in the room. Researchers wanted to know what the baby human would do if they replaced these life-size toys with perfect miniature replicas. How would they physically relate to them?
I'll go for it.
Madeline notices that there is something different about the room now.
Why don't you pretend like it's the big one and you can get in and take it for a ride?
But that doesn't stop her from treating the toys as if nothing had changed. Ignoring the size of the car, she proceeds to try and get into it.
That's a good idea. You should push it in as far as it goes.
At Judy Delocha's lab at the University of Virginia, while studying children's understanding of scale, researchers discovered a very surprising phenomenon involving self-awareness, a crucial step toward becoming a social being.
All right.
2.5-year-old Madeline is invited to play in a room full of large toys. At this age, she already has a sense of herself and would have passed the shopping cart test with flying colors. She's given hands-on experience with each toy in the room. Researchers wanted to know what the baby human would do.
So I'm going to stop that there and just go over a couple of different things. The first thing is we know for filming, you'll hear, if those of you are really astute with experimental design, you'll be like, she tells her to play with it like it's the big toy. Yeah, but in the real experiments, that's not quite what happens. That's just what they seem to do in filming. But this is an example of not being able to quite connect those two streams of information. There's a whole lot of other studies that have done this. So just the inability to connect what an object is and the size of the object, it involves not quite having a very strong connection or any at all between these two streams of information. And the older that kids get, the better they're able to do this and the better they're able to adjust to the difference in the size of the toy and things like that. So it turns out that there are some other clever experiments where we can see that, yes, these two pieces of information are kept quite separate in the brain. And actually, this was not the original intent of this study, was to separate these dorsal and ventral streams of visual information. But it does show an example of what happens because those streams have to develop and process separate pieces of information that eventually need to somehow connect so that we can process what happens in the world around us. So anyway, with that, I will leave you all to think about these things. We come back next class with part 2 of this, and we start to talk about some of the specifics. So what you should walk away with today is an understanding of how transduction happens, the cells that are involved, and then the pathways that are involved, the functions of kind of certain cells, and as well as the connections they make throughout the brain, and then a really basic idea of the information that is processed by each of those. I guess another thing that I should say just very quickly is the fact that we're spending 2 lectures on this should indicate to you that a majority of our quiz coming up would probably focus on visual information. I would say that it's safe to assume that around 66% of the upcoming quiz would focus on information from the visual system, and that means that the other 1/3 or so is going to focus on the auditory and vestibular system. So it probably won't be perfectly that, but you should bias yourself towards understanding all of this visual system information we're going to talk about. Okay. Again, have a great day. Trying to keep these lectures a little shorter. This one's one of the first ones that's pushed to the limit. If you have questions on any of what's going on here, any of the things we've talked about, please, please, please stop in at office hours. I'm here to help. You will feel so much better if you handle any of your questions before the quiz comes up, I promise. Have a great day, and I'll see you next time.